In this episode, we're diving deep into the world of Parkinson's disease with our esteemed guest, Dr. Gagliano Taliun. She's been examining an unexpected intersection, bad cholesterol and Parkinson's disease risk. But there's a twist. Her research probes whether this connection might differ between the sexes. Get ready for an intriguing journey into the genetic intricacies of Parkinson's disease, cholesterol, and the nuanced differences between the sexes. Dr. Gagliano Taliun, I'm so grateful that you're here. Thank you so much for accepting my invitation.
Thanks for having me.
So you did your undergrad in biochemistry and human biology, but how did you end up doing machine learning and statistics?
Yeah, thanks for the question. So I did my undergraduate at the University of Toronto in Ontario, and I really enjoyed my biochemistry courses and also human genetics. But at the end, so in my fourth year, I had the opportunity to do some undergraduate research in a real lab. So this research project was actually doing lab work. We were working on mice, and I was genotyping their DNA, and it was really exciting. But at that moment, I realized lab work was really not for me, and what I really liked was the analyzing part, the statistics and the math. So that's kind of how I transitioned after undergrad to a PhD using computational genetics and machine learning.
Yeah, cool. So we're here to talk about your paper, Lipid-Lowering Drug Targets and Parkinson's Disease, a sex-specific Mendelian randomization study. First of all, can you briefly explain to us what Mendelian randomization is and how it was applied in your study?
Sure. So Mendelian randomization is a causal inference technique that uses genetic variants associated with an exposure. So let's break that down. What does that mean? So first of all, when I talk about causal inference techniques, I like to talk about a clinical trial, so a randomized controlled trial, where you have two groups. You have a group who are taking the medication or the treatment, and the group that's not taking the medication or the treatment.
Right.
You follow them up over time and assess the outcomes, who has disease and who doesn't have disease or lesser. And Mendelian randomization, it actually works in a very similar manner, but the group assignment, instead of the investigator assigning individuals into treatment and non-treatment groups, these groups are predefined by our genetics. So individuals who have alleles or versions of genes that denote a higher level of LDL cholesterol, for instance, as we did in our research paper, they're in one group. And then individuals who have the other alleles, so the other versions of the genes that are not associated with higher LDL cholesterol, they are in that other group. And again, comparing those two groups over time to see if there's a difference in the output. So I guess more practically speaking, Mendelian randomization essentially is testing if there's a causal relationship between an exposure, which is often a modifiable risk factor, such as cholesterol, and an outcome, which usually is a disease.
Cool. So just for my own curiosity, how does it compare with GWAS? Is it something that you do after, before, or doesn't have to do with GWAS at all?
Yeah, absolutely has to do with GWAS. And Mendelian randomization is a type of post-GWAS analysis. So the genome-wide association study statistics are really what empower Mendelian randomization.
Yeah, cool. Can you also explain the difference between the two-sample cis-MR analysis and the standard MR analysis?
Yes, absolutely. So in the paper, we conducted two types of Mendelian randomization. And just to clarify, before we get into the cis versus the standard, there are a broader category of Mendelian randomization called one sample and two sample. So just to make sure no one's confused.
Maybe we can unpack the one sample.
Absolutely. So one sample uses raw data.
Mm-hmm.
When I say raw data, I mean you have a group of individuals, you have their genotypes at positions in the genome, and you have their disease status and their status of the modifiable risk factor, the exposure. And you're using the raw data to do a regression where you regress the genetically predicted exposure onto the outcome, and see if there's a causal relationship between the genetically predicted exposure and the disease. So 1 sample, raw data. 2 sample is what's really empowered by GWAS, the Association Summary Statistics. So in our paper, we use the 2 sample approach, which is where you use summary statistics from 2 different samples. 1 sample for the exposure, so you have a GWAS for your exposure. So in our case, we had a GWAS for LDL cholesterol levels. And you have a second cohort, second sample for your outcome or disease. So we had a second GWAS data set for Parkinson's disease.
So yeah, you were talking about cis versus standard.
Right. So cis refers to nearby, same, similar. And in this context, cis-MR, cis-Mendelian randomization, is referring to the use of genetic variants within a gene. So the target here is a gene. And the reason for cis-MR is that it can be more interpretable. If you are using genetic variants that are associated with your exposure, so associated with LDL cholesterol in a gene, you know exactly what kind of biological function of that gene is. And, you can imagine that this is important for downstream analyses such as drug repurposing and really understanding the biology. For standard MR, instead of using gene variants that only fall into genes, basically you look genome-wide across all of the chromosomes and identify genetic variants genome-wide, regardless of whether or not they are in a gene, to be associated with the exposure of interest. So it's a broad sweep of variants that are associated with exposure, rather than where cis-MR uses a more refined approach, variants that fall specifically into target genes of interest.
And before we move on, can you also explain some of the applications of MR?
So Mendelian randomization has become a very hot topic in recent years. It's become a hot topic for identifying causal relationships between modifiable risk factors and a variety of diseases, you name it. Heart disease has been something that's been studied a lot using Mendelian randomization, and factors such as C-reactive protein levels, so a marker of inflammation in the body, also cholesterol levels and other, basically other exposures where there are medications or drugs already targeted to improve those values. Those have gotten a lot of attention in Mendelian randomization because of the interest in improving health.
You do your MR, and what is usually the next step after that?
So after Mendelian randomization, and with regard to the bioinformatics or the computational side, if someone has done cis-MR, meaning they have looked at genetic variants within genes, and if the result is significant, something that's often done is another approach called co-localization. And in co-localization, the idea is the causal variant for the exposure co-localizing with the signal for the disease. So basically identifying, is it the same signal or is it a different signal but just the same gene?
I was wondering if you could elaborate on the concept of the sex-agnostic approach and why it may not be the best method for this type of study.
Yeah. So sex-agnostic approach, what I refer to that is basically using genome-wide association study results that are derived from studies where men and women are assessed together. The reason why sex agnostic studies may not always be the ideal approach is when you're dealing with a trait, either or both the exposure and the outcome, where there is evidence of sex differences, particularly differences with regard to genetic heterogeneity between men and women. So for LDL cholesterol and for Parkinson's disease, so our exposure and our outcome here, both of them have marked sex differences. So, for example, for Parkinson's disease, it's more prevalent in men than in women. Response to treatment differs between the two sexes, as does the symptoms. So there are some symptoms that are more prevalent in women and others that are more prevalent in men. With regard to the genetic side of Parkinson's, there was a recent study published a couple of years ago where they conducted sex-specific genome-wide association analyses in men and in women separately. And this is actually the publication that helped us conduct our Mendelian randomization. So it was our second sample, the Parkinson sample. In this study, they did not find marked sex heterogeneity in the autosome, so chromosomes 1 to 22. But looking at chromosome X was something that they noted as a future step, and also, of course, increasing sample size to maybe have more statistical power to detect differing effects between the sexes. For LDL cholesterol and other traits similar, so triglycerides and HDL cholesterol as well, there are several locations in the genome where there are differences between men and women with regard to genetic associations. So, for instance, there are certain signals, genetic signals, where the association between LDL and that variant is stronger in women and less strong in men. So there's clearly some dimorphisms happening at the genetic level for LDL cholesterol. So that really motivated us, and also given the clinical differences between men and women for cholesterol and for Parkinson's, to undertake this sex-specific approach to see if we can identify relationships that may be present in men and not in women, or vice versa.
How did you go about investigating the relationship between lipid-lowering drug targets and Parkinson's disease?
Yeah, so in our group, we're very interested in studying the genetic contributors for neurodegenerative disorders. So we decided to focus on Parkinson's here, given the recent availability of sex-specific summary statistics from GWAS. For LDL, we're interested in assessing various pathways associated with Parkinson's. One of them is immune pathways, metabolic, cardiovascular pathways as well. And we decided to focus on LDL to begin with, given kind of the applicability, the transferability. So there are lots of medications to treat LDL cholesterol levels. They target specific genes. We know what those genes are. So on a practical point of view, it facilitates cis-MR, again, more interpretability. And there are very large-scale genome-wide association study results that have been stratified by sex for LDL cholesterol. So basically, it was a combination of interests in our lab and with the data that's currently available that motivated the study.
Yeah, since you mentioned data, can we also talk about the genetic data sets that you used in your study?
Yes. So for the Parkinson's disease study, it was a recent sex-stratified GWAS published a couple of years ago that I had mentioned. And for the LDL cholesterol, so again, in two sample, you have to make sure that the data set for the exposure is a different data set than that from the outcome, or else kind of assumptions collapse. So for the cholesterol, we actually used data from the UK Biobank, which is a very large cohort of 500,000 individuals recruited in the United Kingdom, they're around middle-aged. Around 54% of them are female. So that was our data set behind the exposure.
Mm-hmm. And how does your work fit into the broader context of research into modifiable risk factors for Parkinson's disease?
Yeah, so there is a lot of effort going on for neurodegenerative disorders and, of course, other disorders as well, trying to identify causal mechanisms specifically through modifiable risk factors. Obviously, because that will be key for personalized medicine and improving health outcomes. And for neurodegenerative disorders, I personally think it's really a critical need given the lack of effective treatments for Parkinson's, for Alzheimer's, and for many other disorders that affect mainly the elderly. So this is key, and I feel like a lot of people are working in this domain, not necessarily in the biostatistics computational level, but there's also people who are doing work in animal models, in vitro, in clinical studies, etc. So using a variety of resources to test the possible effects of modifiable risk factors on disease progression, disease onset, and disease outcomes.
Mm-hmm. So although your research didn't find a causal relationship, I was just wondering if you could talk about any surprising or unexpected findings that came out of this paper.
Yeah, thanks. I love this question. So our surprising finding is going to emphasize how nerdy I am. So when one of our algorithms that we used for Mendelian randomization, so there's a couple of techniques that we did, a couple of sensitivity analyses just to confirm our results, since the different approaches, they all have different assumptions, different limitations. So it's standard procedure to use a couple just to check for consistency and make sure things look good. So one approach that we used two computational packages, both in the R language to run this test. It's called MR-EGGER. And we noticed that if we used one package, results were quite different than if we used another package. Mm-hmm. And the nerdy person inside me got super excited about this. So we delved into the code of the two packages, contacted actually the developers of both, had really engaging conversations about statistics and assumptions. We sorted it out in the end, but it was just it was really nice actually to get my feet wet in the code and to really explore further at what was going on.
Mm-hmm. So you encourage future work using causal inference techniques to better understand sex-specific effects. Can you expand on what kind of research you hope to see in the future and how it might help us better understand the complex disease risks that vary between sexes?
Yeah, thank you. So in my group, we're very interested in precision medicine, and I believe that sex-specific analyses is a key way of moving towards precision medicine. So currently in the clinic for a lot of diseases, differences between men and women are not always accounted for, and this can lead to bias or inequitable care in these groups. And my goal is really to incorporate sex-specific analyses to try to help fill this gap, to try to move towards precision medicine that is targeted differently between men and women. So taking into account genetic and underlying biological differences to really improve health outcomes for both.
Mm-hmm. So I was also wondering how you use machine learning in your group.
So machine learning is one of the approaches that we use to answer and to move forward in the question of using sex-specific approaches. So one project that we currently have in the group is developing sex-specific prediction models. Currently, these prediction models are for coronary artery disease. Mm-hmm. So what we're doing here is we're currently trying to get a baseline. So we're taking several different machine learning models and deep learning models. So each have their own assumptions, each have their own advantages and disadvantages, and trying to get a baseline of which model performs best, and which model is most computationally intensive, which is most robust. So we're doing this in the UK Biobank dataset. So the same dataset that we used for the LDL cholesterol values. The reason why we chose this dataset is simply because of the vast size. So for machine learning, your training set, it's critical to have large sample size, a lot of different features, and to improve the training. So UK Biobank is what we chose for that. And currently our investigation is combining both non-genetic, so environmental, clinical factors, in addition to genetic variants throughout the genome to develop these sex-specific models for coronary artery disease. Again, coronary artery disease, another disease where there are quite striking differences in outcomes in response to treatment, etc., between men and women. So an ideal setting to test these models. So this work is ongoing, but we're really excited about it. And the ultimate goal is to, in the end, have models that are specific to women, specific to men, that can be used to help identify individuals who are at a higher risk of developing coronary artery disease. So interventions can be done sooner and they have longer, healthier lives.
Mm-hmm. And I'm just wondering, as a researcher, what is the algorithm that you have used the most so far in your whole career, a machine learning algorithm?
Yeah, so it really depends on the question you're asking. We always do like to start with the so-called basic machine learning models. So for instance, a regression. So a penalized regression where you have penalties applied to a normal regression is a fairly simple form of machine learning. But I feel simple isn't always a bad thing. If you can accomplish the same task with less computational resources and a more interpretable model, that's a pretty good thing. So we usually start with some type of penalized linear regression. Random forest, so a collection of decision trees, we often also use, again, for interpretability. There are caveats and issues with both of those methods, but I feel that's the benefit of employing many and seeing if we can create some sort of ensemble approach, so an approach that takes advantage of the benefit from the various methods to create an optimal model. For deeper machine learning, so less standard machine learning techniques, currently for the questions at hand, we're using a multi-layer perceptron. The reason why was really due to the data. So we don't have imaging data where you would probably use a different approach. We're not currently using longitudinal data, again, where you would probably also use a different approach for that. But yeah, I'm a real advocate for trying multiple methods, and the more complex may not necessarily be the best answer. And that's the joys of research, is to explore and figure out what is the optimal model.
Mm-hmm. Yeah, and as someone who switched from wet lab to dry lab, how did you learn programming and all of that yourself?
So I learned programming while doing my PhD at the University of Toronto. So it was really hands-on experience. For the Research Alliance of Canada, formerly called Compute Canada, they held summer workshops and workshops throughout the year. I think I joined maybe every single one of those. But it was really practical experience and trial and error. I also should say I had a lot of colleagues in the lab and in other labs at the university who were really key at helping me if I had any questions. So this environment was also quite ideal. It was a bit of a learning curve, but I was highly motivated. I knew this is what I really wanted to do. So it was an enjoyable learning curve.
Do you also from time to time miss wet lab?
No, And that's a simple answer, but actually it's not at all. I don't regret it. I feel like having that experience, it was definitely a benefit. I have a greater appreciation of the genotypes that I receive, having that experience in the wet lab. But it wasn't for me for sure.
And since we're talking about machine learning, how excited are you as a researcher about all these new language models and like all these cool stuff that you can do with them?
Yeah, I think it's a very exciting time. I just want to make sure that people are using them in ethical ways, and that's my main concern. But I think the benefit of machine learning is that it can help with getting tasks done in an efficient way if done in a ethical manner and a transparent manner.
Thank you so much. I think that was great. Let's wrap up with this question that I want to start asking each guest since a previous episode. So in your journey, what has contributed the most to your success?
Yes, I was looking forward to this question. For me, there are several things. The first would be having balance. So I feel like for me throughout my career, I've always been a type of person who has a lot of hobbies, likes to do things outside of the computer. That being said, I 100% find joy in my research. I wouldn't be in my current position if it didn't make me happy. I'm a strong believer in doing what makes you happy. But I feel like having a balance of doing external other things, it just makes the research that much more enjoyable. So in my spare time, I like to write a lot. Of course, I'm writing a lot of scientific articles, but I'm not referring to that. I'm referring to other forms of writing. So I've written several, I think 3 articles that have been published in Nature about basically career things. So kind of my experience in academia with regard to teaching, with regard to finding a faculty position, etc. So that brings me a lot of joy writing. I also do writing that's completely non-scientific related. So I have like an article I wrote about my trip to Paris and my trip to London, things like that. I really enjoy writing, and then it's a nice balance with the research. And I guess for advice I would give individuals who are in graduate school or thinking about continuing in academia is to really make sure that your underlying reason of why you're doing grad school or why you're pursuing research comes from a good place. So meaning you personally, this is what you want, not what someone else wants or not what someone is expecting from you. I feel that when it comes from the right place, it's really enjoyable and you get a lot more out of it.
Yeah, I completely agree. Thank you so much again for showing up. I think that was great.
Thank you.
All right, I really hope you enjoyed this episode. If you did, please leave us a great review on your favorite podcasting platform, and I'll see you again next time.